Now it's my real special pleasure to announce Professor Christine Klein for the David Marsden Lecture, and she deserves this award for various reasons. She made outstanding contributions to the field of neurogenetics, of dystonia and Parkinson. She initiated the MDS gene tool here in the society. She was active for many years in that field. She published over 400 scientific peer-reviewed articles, and she is involved in so many collaborations all over the world. I think many people here in the room will know her and may have some small collaboration with her. And currently she's Schilling Professor of Neurogenetics and Neurology. She's director of the Institute of Neurogenetics at the University of Lübeck, and not to forget, a lot of work, she is the current president of the German Neurological Society, the first woman of that society as a president. And this is also a congratulation to her because it happened just this year. And we are really looking forward to your presentation, Christine.
Thank you very much for the wonderful introduction. Dear President-elect of the Society, dear colleagues, I'm humbled by the tremendous honor of having been selected as today's David Marsden Lecturer. And I would like to express my sincere gratitude to all of you, to the Society and to the Program Committee for this award and this outstanding opportunity. I would like to talk to you today about a topic that is very close to my heart, and that is reduced penetrance of hereditary movement disorders. And that is that a certain person, despite carrying a pathogenic mutation, remains healthy or develops the disease only very late in life. And I think this is a fantastic opportunity for us to try and elucidate mechanisms of endogenous disease protection. And Professor David Marsden continues to be a role model for all of us here in the Society, and I'm certain that he will do so and inspire us also in the future. Okay, so I will just try to advance. Okay, here we go. So before I will focus on reduced penetrance, I would like to come back to the special connection of the Movement Disorder Society to Hamburg that we have already heard about, and also to my own personal encounter with Professor David Marsden, because I'm actually old enough to still have had the privilege of meeting him in person. And so you've seen this slide before, and I'm grateful to Professor Fahn who shared it with me, and this is actually his hotel room at the Atlantic Hotel in Hamburg, northern Germany, where in 1985 the Movement Disorder Society was founded. And on the very right, you can see Professor Marsden. Is there any other way, or maybe the battery is a bit... So this is the MDS historic site, the Elegant Atlantic Hotel in Hamburg. And I would like to draw your particular attention to this part of the hotel, which will become later important at the very end of my talk. I don't know if this battery is flat. It's very difficult to advance the slides. So as I mentioned, there is a very special MDS Hamburg connection as the society was founded in this city. I personally also have a special connection to Hamburg as I was born in that city. And here you can see me walking just about a kilometer away from the Atlantic Hotel. And again, this location will also become important at the end of my talk. And today there is a car rental agency. In 1985, when the society was founded, I was still a high school student. Do you think that somebody could help me with, it's very, very hard to do. Sorry about that. And so, but then, 10 years later, at this time I was, This will be working? Okay. Okay, good. I'll try that then. Thank you. Thank you. Thank you. So 10 years later, I've had the privilege to go as a final year medical student to Queen Square, London, where Professor Marsden at the time was walking, and I was admiring the grand rounds that he gave. And so this is my time at Queen Square, and I also became very interested in research and in movement disorders very early on, and as you can see here, and I was supervised actually by Neil Quinn, and one of the efforts that we undertook there resulted in a small paper published in the Movement Disorders Journal entitled Ataxia Without Telangiectasia, Masquerading as Benign Hereditary Chorea, where Professor Marsden is the senior author and supervised me and that work, and I am obviously very honored and grateful for that opportunity. So now let me focus on reduced penetrance. And first of all, I would like to introduce you to the concept and to the translational impact that it may have. I will cover some genetic modifiers that have already been identified, turn to human studies and models, and then finish off with future directions. So, as I mentioned before, reduced penetrance refers to the fact that a confirmed pathogenic mutation does not manifest. And I would like to extend that concept to the fact that it could manifest late or later in life. So what keeps a non-manifesting mutation carrier healthy? And I think this is a very positive way of looking at the coin, where you have risk factors and protective factors, but it is not exactly the same, because when you want to study and when you want to learn about protective factors, you also have to look not just in affected individuals, but also in healthy people. So this is a little girl with myoclonus dystonia, one of the first patients that I saw when I came back from the United States. And she has a condition that you may know is a very good example of a condition with reduced penetrance. And it was Katja Luhmann's thesis where she was able to identify why that is the case, that not all of the patients manifest the disease. And that is because the epsilon sarcoglycan gene is paternally expressed and the maternal allele is imprinted. So this is an epigenetic mechanism explaining reduced penetrance. Let me go into a bit more detail about the concept. So there are many facets, I find, of reduced penetrance, and I would like to extend, as I mentioned before, I would like to extend that concept beyond a simple black and white distinction. And so first of all, however, let's look at this all or none phenomenon, and this is true for the SGCE gene in myoclonus dystonia. And you can see here an example of a pedigree from Canada where several generations are being spared, and when you look why this is the case, it's the case because the mutation is always inherited from the mother to the next generation, and only when the mutation is inherited from the father in the last generation in the gray box, only then the children manifest the disease. As I mentioned, penetrance is often age-dependent, and this is even true for recessive conditions with high penetrance. And you can see here two brothers that were taken care of by Peter Braunstahl and Balzano, and they both carry a Parkin mutation in the biallelic state. And one brother, the one on the left, however, developed the disease at the age of 32, whereas the other brother only developed the disease at the age of 58. So there can be very, very large differences in manifestation in penetrance, even within families carrying the same mutation. This is perhaps even more striking in dominant diseases, and I think a very good example is this work from Matt Farah and Joanne Trin and Faisal Hentati, where they looked at LRRK2 mutation carriers. All of these people carry, and these patients carry, the same G2019S mutation. However, the age of onset and the penetrance is very, very different in these different populations. So if you draw a line here at the age of 60 and you look at the Norwegian cohort, you will see that the incidence of Parkinson's at that age is only about 20% in that population, compared to the Arab Berbers, where at the same time, at the age of 60, already about 60% will have manifested the disease. And this obviously suggests either genetic factors or environmental factors or a combination of both that will shape this penetrance. And finally, I would like to also point out, and I'm sure you're all aware of this, that really penetrance abuts on variable expressivity, and we have to take good care, and we just heard a beautiful talk about this, we have to really look very early on in prodromal phases and have to look for endophenotypes. And I would like to illustrate this here again with an example. So this is a young lady who came to us to ask whether there's anything wrong with her motor function. And I'm sure you would all agree with me that she's absolutely fine. However, her father carries a LRRK2 mutation, is affected with Parkinson's disease, and she knows that she is carrying the exact same mutation. We performed transcranial ultrasound in her, and as you may know, there is, and I will encircle this here, in the brainstem, in Parkinson's disease patients, in about 90% of the patients, you will find hyperechogenicity in the substantia nigra. And this is very clearly the case in our person here. I wouldn't call her a patient, but so there may be something going on in her brain, and it will be very important to study these findings in a longitudinal manner. And how important that is, I would like to illustrate with another case. So this is a gentleman with a heterozygous PINK1 mutation, and I'll come back to the role of heterozygous mutations in a minute. And so he showed in 2007, so 12 years ago, some early signs of Parkinsonism. Of course, not enough to make a diagnosis of Parkinson's, but I'm sure you'll agree that there is something wrong with his arm swing, and you can see this even better when he walks. And there was also some slight rigidity in that same arm. And so we were thinking and expecting that he may in the future develop more signs of Parkinsonism and go on to progress. We have just seen him again, and this is Dr. Balk examining the patient 12 years later, a few weeks ago, and here you can see him. He gained a few kilos of weight. Other than that, he still doesn't swing his right arm. It's a bit slower perhaps, but I don't think it's due to, he has had several operations, and so I don't think it's due to Parkinsonism. And if you see him here moving, for example, the tapping movements, I think this is perfectly fine, and really we could not establish a diagnosis of Parkinson's, nor could we really see any progression. So I think this is very important as well. So there seem to be individuals, despite having subtle signs or endophenotypes, that never really progressed to Parkinsonism. And again, this will be very interesting to study what is different in these individuals from those who go on to develop the full-blown disease. So there are many mechanisms one can think of, and I'm sure you can think of even more. So one could be the mutation type, another one could be modifier genes, and I'll talk about that. There could be expression levels, there could be epigenetic modifiers, as we saw in the case of myoclonus dystonia. Obviously, the environment will play a role as well, although that's more difficult to study as we don't have the environmentome available as we do have the genome now. And there are, of course, intrinsic factors, and they are very important, such as age and gender. So let me come now to the translational impact. And I would like to turn back to the little girl that you saw at the very beginning with myoclonus dystonia, who is now in her early 20s. And she still has myoclonus dystonia, and she has become very interested in the disease. She has written a thesis on her condition, and she's obviously becoming interested in her own life, career planning, family planning. So I was able to tell her that the SGCE gene is maternally imprinted, and really, even in the absence of a complete understanding, complete mechanistic understanding, we can impact clinical care through findings such as the imprinting mechanism in SGCE. So I could tell her that the mutation will most likely, even if her children do inherit it, and they have a 50% risk, even if they do inherit it, her children will have almost zero risk to develop the disease, which is obviously an important piece of information. Let me now turn to genetic modifiers. So first of all, let's review what the penetrance of different movement disorders genes is. And I have plotted here on the y-axis the penetrance in percent, and on the x-axis you'll see the frequency of the different variants and mutations. So let's start out with the rare recessive movement disorders genes, such as Parkin and PINK1, who tend to have a very high, albeit age-related penetrance, as we saw. And then let's look at TAF1. So TAF1 mutations cause X-linked dystonia Parkinsonism. So this is an X-linked disorder, also with high penetrance, but I'll come back to that as well. Here you can see heterozygous conditions. So these are genes LRRK2 and VPS35 that cause dominant Parkinsonism. And here, as you can see, penetrance is quite markedly reduced. And this is a bunch of dystonia genes, here all also heterozygous mutations, all dominantly inherited, all with reduced penetrance. And then finally, and this is a very interesting observation, and I'm sure you know about this, heterozygous mutations in otherwise recessively active genes seem to be able to predispose or to act as a risk factor to develop, for example, Parkinson's disease. And really this scheme shows you that it is very difficult, if not impossible, to make a clear distinction between a strong risk factor and a gene, a dominant gene, for example, with highly, highly reduced penetrance. So I would venture to say that there really is a continuum. And I will now focus on these two genes, Parkin and Pink, TAF1 and TOR1A, as examples. So what are these genetic mutations you want? This was a beautiful cover a few years ago, PNAS, so just have the right umbrella, right mutation that protects you. So let's look into this, and let's start out with a relatively historic example. So this is the TOR1A gene, and you know that the GAG deletion causes early onset generalized dystonia. And with reduced penetrance. So penetrance is anyway reduced in this condition to 35%. If you carry on the other allele a rare, it's rare, so a rare polymorphism, then in combination, your penetrance will go down to 3%. So almost completely protected from developing the disease. I should say, and this has been replicated in this study by Kam and colleagues in 2008, and I should say there is one study that is sometimes reported as refuting this finding, and that's not true though, because in that study simply there was no carrier of the D216H polymorphism, so the authors couldn't test it. So it doesn't refute it, so this has never been refuted, and I think it would be important to try and replicate this and potentially also even include this into genetic counseling. I would now like to turn to X-linked dystonia Parkinsonism, a very interesting model basal ganglia disease that is endemic to the Philippines, to the island of Panay, and it is caused by a retrotransposon insertion in the TAF1 gene. Here you can see a patient with the typical combination of dystonic and Parkinsonian features as seen in these patients. And due to a wonderful collaboration with our Filipino and also with our Boston colleagues, we were able to do some research into genetic modifiers of this condition. First of all, the Boston group found that within the insertion that I mentioned as being causative, these patients, in addition, have a hexanucleotide repeat expansion. So that was a very interesting finding, and this hexanucleotide repeat expansion within the actual mutation does have a very large impact on the age of onset in this relatively highly, probably very highly penetrant disease. And we were able to confirm this observation in a very large number of patients, almost 300 patients, and here you can see a very nice correlation between number of repeats or repeat length with age of onset, and obviously this is something that we know very well from the repeat expansion disorders. The effect is so strong that we were even able to see it in a much smaller cohort of about 20 patients where we had additional information on specific features of the disease, such as, for example, cognitive function. We were then interested in the brain region specificity of the repeat length. And for this, we performed small pool PCR analysis where really you're trying to look at single molecules followed by Southern blotting. And I would like to share those results with you. So here you can see of one individual, we have performed Southern blotting of different samples in blood. So these are all from the same individual, and you can see that the repeat length is the same in pretty much in every single lane of the blot. When, however, looking at the brain of this individual that was available to us, then you can see here that in the basal ganglia, as one tissue that we examined, there's quite marked mosaicism. And also, not only that, but also the repeat length is larger, is longer in cortex, for example, and in the basal ganglia. than in blood. So this is very interesting from a perspective of disease pathogenesis, but also from a penetrance perspective. And we had a second patient available at the brain from a second patient. This is shown in panel C. And again, you can see from various brain regions that there is a variable number of repeats, again with cortex and also the basal ganglia standing out as having the largest repeat numbers. I would now like to share some additional unpublished data with you because this age of onset modifier that I just showed you for XDP, the repeat expansion, cannot be the only one because it explains only 50% of the age of onset variance. So we set out to try and find additional age of onset modifiers in this disease, and for this we performed an age of onset Geos analysis, Genome-Wide Association study. And we had at that time available to us, together with the Boston group, 344 unrelated XDP patients. As you know, this is an incredibly small number for a GWAS. And but we said, let's try. And so the age of onset was about 40, with a range of 20 to 67 years. We corrected, of course, the GWAS for repeat number and also performed fine mapping. And what we found, you can see here on the Manhattan plot, we found, to our great surprise and great pleasure as well, we found two regions that were highly significant at the genome-wide level, quite unexpected given the small number. And what is particularly beautiful is that these two hits are very interesting in terms of the pathophysiology and make a lot of biological sense. So we think that we have found another important modifier here for X-linked dystonia Parkinsonism. Let's shift gears and turn to mitochondrial Parkinsonism. As you know, the prototypes for this are Parkin and PINK1 related disease. And to quote Enza Maria Valente here in her paper when she discovered PINK1, these data provide a direct molecular link between mitochondria and the pathogenesis of PD. So let me show you some of the work of Joanne Trin. So first of all, she performed deep mitochondrial sequencing using next-generation sequencing technology, 10,000x deep sequencing in samples from controls, heterozygous PINK1 mutation carriers, kindly provided by Matt Farah and Faisal Hantati, and PINK1 homozygous carriers. And you see a nice stepwise increase in the number of mitochondrial variants, total count, across these different gene dosage genotypes, if you will. How about penetrance? So indeed, it looks like mitochondrial DNA mutations might also influence penetrance. And if you look, please, here at panel B, you will see a comparison for Parkin. On the left, you see Parkin mutation carriers that are negative for PD, so that don't manifest the disease, compared to those Parkin mutation carriers that do manifest the disease. And it seems like the manifesting people actually have a higher mutational load, a higher number of heteroplasmic mitochondrial DNA variants. And the same is true, and this is shown in panel D, for PINK1. So we thought this was an interesting finding. Another interesting link between mitochondria and inflammation has recently been published in this paper in Nature, and it elucidated the pathway of how sting-induced inflammation can be triggered by the release of mitochondrial DNA in patients with, and well, first in mice with Parkin and PINK1 mutations. And so this is the data here. The mice Parkin in blue, PINK1 in orange, and you can see highly elevated levels of different cytokines, interferon beta-1 and IL-6 here in those mouse groups. And interestingly enough, a strong, not as strong, but also a very significant elevation was present in heterozygous mice and also in a pilot study in humans. So we next set out to try and validate and replicate and validate these findings in larger cohorts, and this is data from a paper that we recently submitted, Max Borsch and colleagues, and you can see here that indeed the IL-6 levels, we focused here on IL-6 as a cytokine, IL-6 levels are the highest in mutation carriers with 2 mutations, biallelic mutation carriers, and Parkinson's present, compared to mutation carriers of just a single mutation with PD, mutation carriers without PD, and healthy controls. And the nice observation here in the Italian cohort, kindly provided by Enza Maria Valente, is that we were able to replicate this finding in a totally independent cohort as well. I would now very briefly like to touch on human studies that one can perform in order to understand penetrance better. And I would like to first of all start out with a very old study. This is the first one we undertook 18 years ago. And this was done in the family where I showed you the 2 brothers with Parkin mutations at the beginning. So in this family, we were able to undertake a PET study, fluorodopa PET and raclopride PET study, and you can see that for both PET markers, there is on the very left you have the control subject, and then the next images show those from asymptomatic heterozygous Parkin mutation carriers. And you can see, compared to the compound heterozygous, so the ones with 2 mutations, that they really fall in between what you see in controls and in the biallelic mutation carriers. So at the PET level, very clearly, there was also something visible already in the heterozygous carriers. Another more recent example that I would like to share with you here is again from the field of X-linked dystonia Parkinsonism. And this is a mutation carrier, again, who came to us in order to participate in a research study. And he's perfectly fine when he walks. He's also fine when he sits. And when, however, he's wiggling his tongue from side to side very quickly, you can see that this is certainly not normal. And also when you look carefully at his 4th finger on the right hand, there's a little bit of a tremor or a twitching. And so there may be some very subtle signs, clinical subtle signs in him, but you really have to look very closely. So he also underwent an imaging study, in this case MRI imaging. And this is an N-of-1 study, but I would just like to show this to you as an example as to how we may be able in the future to place individual mutation carriers in a very detailed and hopefully very accurate manner at the different stages where they're at. And so in this disease, in X-linked dystonia Parkinsonism, the anterior medial putamen is affected first, and only later the dorsolateral putamen is going to be affected. So on the left, you can see the values for controls versus patients in red, and in the middle, just the one bar indicates our mutation carrier. And so you can see for the anteromedial putamen, he falls in between the controls and the patients. So there's already something going on in his putamen. However, when it comes to the later disease stage marker, the dorsolateral putamen involvement, you can see that he completely falls still within the controls. One word on models. Obviously, there are many, many different models, cellular models and other models that, apart from the human models that we obviously also study, models that can be used. And one model that we have been using, and I cannot show you the results yet, but I can at least show you how we're trying to tackle this, is based on Drosophila. And we came back to a study published a number of years ago in Science, where vitamin K2 was found to be and to act as a mitochondrial electron carrier that can rescue PINK1 deficiency. And for this, Melissa Voss and her colleagues used a PINK1 knockout fly. And when I looked at this data from this paper, and Melissa has now joined our group in Lübeck, when I looked at this, I said to her, well, this is interesting, but 10% of your flies can still fly. So what is it? This is a penetrance issue. So what do these flies do better than the flies that can't fly? And so through a series of very elegant experiments and Drosophila crossings, Melissa has now generated all of these offspring. And as you can see, there is a very broad variability when it comes to flying capacity. And so these are the flies that cannot fly at all. And here are the flies where over 50% of the flies can fly. and as a readout for Parkinson's, I should say. So what we're doing at the moment is we're trying to perform a modifier screen in Drosophila and are undertaking a transcriptome analysis by looking into these extremes of flies in the hope that we will be able to find modifiers. And we do have several interesting candidates that we're currently validating, and then we'll compare to what we find in humans. So I'm now coming to the end, to the future directions, and would like to start out by sharing with you a study that at first glance may not have much to do with movement disorders at all, and it really doesn't. So this is a paper that I was very intrigued by that was published in June in the New England Journal of Medicine. And in this study, patients, or no, I should say people, and in this case these were even children with an average age of 14, were given an anti-CD3 antibody for 2 weeks. And so these children all had to be at risk to develop type 1 diabetes. And so they had to have 3 different risk factors that had to be present, including a first-degree relative with diabetes type 1, a problem already with glucose intolerance, but not allowing for a diagnosis of diabetes, and third, antibodies indicative of later development of diabetes. And so this reminded me of what we've just heard of the different scores of the prodromal work that we are undertaking in Parkinson's, that perhaps it could be possible to also identify people very early on in the future. This will be years down the road, but it may be possible to treat them very early on. And so this trial was very, very successful, and I hope it will replicate, because here just the 14-day course of this antibody resulted in a 2-year delay of the development of diabetes in the children receiving the antibody compared to the placebo group. Yeah, so when trying to tackle reduced penetrance, obviously we need mutation carriers. Obviously this has to be a global effort, and only through this we can get at the numbers that we need. And I would just like to highlight one huge collaboration that we've been embarking on since last year, and many of you here in the audience have generously contributed to this. Over 100 centers have contributed data on mutation carriers. And initially, we were reported over 8,000 mutation carriers, and now we have exact data from 4,500 mutation carriers. So this is a wonderful contribution, and I would like to thank everyone here for doing this, and I look forward to advancing this collaboration, hopefully to learn more about penetrance in the future using this resource that is also supported by the Movement Disorders Society. It's built on the MDS gene effort, the GOPD initiative, and funded by the Michael J. Fox Foundation. So I would like to now come to my thank you slides, and first of all, I'd like to mention my mentor in Lübeck, Detlef Kempf, and then Neil Quinn, who encouraged me to perform research at the time when I was still a final year medical student at Queen Square. I would like to thank Sandra Brakefield, who taught me genetics in Boston, and Tony Lang, who taught me movement disorders in a series of 11 short-term research visits and later sabbaticals, and last but not least, my family, who bear with me all the time. And I would also like to thank the Movement Disorders Society, that has been supporting me over all these years and inspired me. And I would just like to mention one recent specific piece of support, and that is a grant that we got to support this conference here. So this is, to my knowledge, the first conference on reduced penetrance, or it was the first conference. It was held in 2018 in December, and we had 10 outstanding speakers contributing to this conference and over 100 participants, and this was co-funded by the Movement Disorders Society. Finally, I would like to come back to the team. The only reason why I'm standing here is obviously that all of this is a tremendous team effort over 20 years, and not only with my own group, but also with the collaborators, with the funders, and with the probands who so graciously generously donate their time. And so I would like to thank all of them. And finally, I would like, and now for this very last video, I would like to request sound, please. I would like to show you one final video, and I think we're now coming all full circle. I hope you will agree. And you may be not aware of the fact that there is even an example of reduced penetrance in the movies. And that I would like to now share with you. And just remind you, this is the Atlantic Hotel, where the Movement Disorder Society was founded. Here you see the globe, the big globe at the corner that I showed you before. And this distance from the hotel to this place where the car rental agency is will become important in the video. So let's look at the video, and please give me some sound.
Congratulations on a safe journey.
So with that, I would like to thank you again and finish and wish all of us a safe journey, and tomorrow never dies.
Quite charming. Thank you for the wonderful presentation.
Thank you.
So I think we had a really fantastic end of that session. And I think I have nothing to add here. And I thank you for coming, and I wish you a successful and fruitful Congress. Thank you.